Hello everyone. Today we are back with the biological classification in which we are going to read about the 5 kingdom classification proposed by Robert Whittaker. Robert Whittaker classified all the living organisms into 5 kingdoms, and these were kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae and kingdom Animalia. Today we are going to read at first about your kingdom Monera. Now, kingdom Monera comprises solely of all the different kinds of bacteria. Okay, so the members of the kingdom Monera are all bacteria. Now, this bacteria has been further classified by that of your Carl Woese into 2 groups, and those were your archaebacteria and that of your eubacteria. But along with that, there is other kind of organism also which has been included in the kingdom Monera, that is called as your mycoplasma. Now, what are these mycoplasma or why it has been included in kingdom Monera, that we will be reading one by one. So, let us first see the characteristic features of the kingdom Monera, which include the eubacteria, archaebacteria and the mycoplasma. Now, the bacteria are ubiquitous in nature. What do you mean by this word ubiquitous? That means they are found universally almost everywhere. No place on this earth is free from bacteria. Whether you go to the hot sulfur spring, whether you go to the desert, whether you go to deep inside the ocean where there is no oxygen concentration, you will find everywhere bacteria is residing. As far as this classification of the bacteria into different group is concerned, that is primarily based upon the fact that the eubacteria and archaebacteria varies in terms of their cell wall and cell membrane composition. The cell wall of this bacteria, that is the members belonging to that of your Monera, except mycoplasma, because mycoplasma does not have cell wall. If we talk about the eubacteria or the archaebacteria, their cell wall is not made up of cellulose. In case of plants, the cell wall is made up of cellulose, but in case of the bacteria, that is eubacteria and archaebacteria, it is made up of a compound, or it is made up of a polymer which is called as your peptidoglycan. So, eubacteria has a cell wall which is made up of peptidoglycan. That means it is a polymer of carbohydrate with that is your protein, and they are cross-linked by the help of certain chain, that is a branch, small chains of your lipids. So, that is the structure of that of your cell wall of that of your eubacteria. And as far as the archaebacteria is concerned, their cell wall is not made up of peptidoglycan. It is made up of yet another compound, and that is called as your pseudopeptidoglycan or pseudomurin. Okay, so this is the fundamental difference between that of eubacteria and archaebacteria, depending upon which they were classified, or they were separated from each other. Also, it is because of this composition of the cell wall of the archaebacteria, and of course the cell membrane is also different from that of eubacteria. It contains, of course, it contains the proteins, it contains the lipids, but the arrangement is different. They have ether bond between them. So, as a result, they can withstand a harsh environmental conditions other than that of the eubacteria, which cannot. So, archaebacteria are capable of living in such a harsh conditions, like they are known as your thermoacidophiles, they are known as your halophiles, they are known as your methanogenic bacteria. So, why have they been given this kind of name? Is because of the difference, or because of the rare combinations which are found in their cell wall and cell membrane, which makes them yet you can say the primitive, but yet they are able to survive in this changed environmental conditions, which has took over thousands and thousands of years till from the formation of the earth till today. So that's why sometimes this archaebacteria are also known as the living fossils. Okay, because they are capable of surviving all the kinds of conditions right from the primitive time when the earth was formed till date. Okay, so this is how the eubacteria and archaebacteria are differentiated in terms of their cell wall composition, and that is why the reason is that archaebacteria are able to live in the harsh environmental conditions compared to that of the eubacteria. Now, this word eubacteria means true bacteria. Now, why they are called the true bacteria? The reason is because the cell wall is made up of the peptidoglycan layer. Okay, so that is the criteria for the true bacteria. bacteria, and also these bacteria are unable to live in the extreme conditions of temperature. They generally are simplified organisms, but they have different metabolic activities or pathways. Some of them are photosynthetic, some of them are heterotropic. That means some of them, those who are photosynthetic, they can be photosynthetic taking the sunlight as a source of energy, and some of them are chemosynthetic bacteria which belong to this eubacteria, which take the inorganic compounds like the nitrates, compounds of nitrogen, nitrates, nitrites, ammonia, to be the compounds which will be broken down to release energy to utilize them for doing their various kinds of metabolic activities. So chemosynthetic bacteria are the one which utilizes this. Then there are certain sulfur bacteria which utilizes hydrogen sulfide to be the source of energy. They break down that hydrogen sulfide. Some are photosynthetic bacteria, so they have chlorophyll in them, but this chlorophyll, what matches with the normal higher plants, that is the normal terrestrial plants, green plants, is that chlorophyll A is common with that of the higher plants. But other than chlorophyll A, they have some other pigments. We call them as your xanthophylls, carotenoids. So these are the various pigments which are associated with the process of photosynthesis in case of your photosynthetic bacteria. Now these photosynthetic bacteria are sometimes known as your cyanobacteria. Now they were also earlier known as your blue-green algae, but but now they are known as your cyanobacteria. The reason is because the algae is a unicellular eukaryotic organism, whereas the cyanobacteria are unicellular prokaryotic organisms. That means their cell wall composition matches with that of the organisms belonging to the Monera. Then they do not have membrane bound organelles. The organelles are just formation of very simplified organelles are there in their body. They have the ribosomes, which is 70S type. Okay, so these are the various fundamental things which makes them classified into Monera rather than being taken to the algae, which earlier they used to be. Now the organisms which belong to eubacteria, they, depending upon their shape, can be classified into 4 types. Now what are they? Rod shaped. Rod shaped are called bacilli. Okay, then round shaped, they are called as your coccus. Spherical or round shape, they are called as your coccus. Then comma shaped, it is called as your vibrio. And then spiral shaped, like that of screw. On the screw, body of the screw, you might have seen that how the spirally the thing markings are there. So spiral shaped, they are called as your spirillum, and the plural is spirilla. So these are the various 4 kinds of shapes found in case of eubacteria. They grow very well at a temperature of 37 degrees Celsius, which is the normal temperature. And they are heterotropic also, as I told you. That means they are either saprophytes, that means they decompose the dead, or they derive the food from the dead and the decaying organic substances. And also they can live on the body of a living host and derive nutrition from them. So they are capable of, they are wide range in terms of obtaining their nutrition. Some of them are autotropic, chemosynthetically autotropic or photosynthetically autotropic, and some of them are heterotrophs like this. So this is the various things. Again, the bacteria can be classified. I'm talking about the eubacteria. They can be classified on the basis of Gram staining. There was a scientist, his name was called Christian Gram, and he actually classified the bacteria into 2 groups, Gram positive and Gram negative. What he did is that he took a few stains and some reagents to prove that which one is Gram positive and which one is Gram negative. That was crystal violet, iodine solution, and either acetone or ethanol. So what he did is that he just made a slide of this bacteria, any kind of, whether it is Gram positive or Gram negative, unknowingly, of course. If he knows already beforehand that which is Gram positive and which is Gram negative, then what is the use, isn't it? So he took a sample of bacteria, he made a slide out of it, added this crystal violet and iodine solution. He waited for a few minutes. After air drying it, he then washed it first with water and then with alcohol or acetone, and then he observed the thin film of that bacteria under the microscope. So he noticed that there were certain group of bacteria which retained the color of this crystal violet, and he named them as Gram positive bacteria. and there were certain bacteria which on washing it with acetone or this ethanol could not retain this color of crystal violet and iodine solution, and they were appearing to be hyaline, that means colorless under the microscope. So if we later on add saffron into it and wait again for some time after air drying and observe it, then we can observe that these group of bacteria could take the color of saffron which was red. So he named them as gram-negative bacteria. One such gram-positive bacteria is your Streptococcus. So Streptococcus is a gram-positive bacteria, Lactobacillus is a gram-positive bacteria, and a gram-negative bacteria, if you talk about E. coli, that is Escherichia coli, which is found in the gut of human beings, that is a gram-negative bacteria. So depending upon the staining mechanism, bacteria can be classified into gram-positive and gram-negative. Then bacteria in normal favorable conditions multiplies by means of asexual reproduction, that is by means of binary fission or multiple fission. But if the situations are unfavorable, in that case the bacteria multiplies by means of formation of endospores. They form a cyst. That cyst is a hard outer covering, a capsule, and within which the cytoplasm and the nucleus undergo divisions. First the nucleus followed by the cytoplasm, and they round off to become some spore-like structures which can resist the unfavorable environmental conditions, and when the favorable conditions again comes, this that is the cyst breaks open and the spores are liberated into the surroundings, which can be taken by wind or water, and wherever they will fall, they will again form the bacterial colony or the unicellular bacteria or the filamentous bacteria as they might be. Okay, so this is how they can reproduce. Sometimes bacteria also show transfer of genetic material from one bacteria to another. Now this is a kind of, since it's a transfer of genetic material and combining it with a new bacterial cell's chromosome, because bacteria has a single chromosome. Its DNA it's single DNA, it's lying freely within the cytoplasm. It is not bounded by any nuclear membrane. So that is why they are prokaryotic organisms, and they do not have histone protein. When you will be seeing the other classes, you have read it in class 10 also, that the chromosome is made up of DNA and histone protein. So in case of bacteria, they do not have histone protein, they have only the DNA. So this DNA is single and it is circular, it is round. So sometimes along with the DNA, they have some extra chromosomal DNA, not all but some of them they have it, and this extra chromosomal DNA can be transferred to another bacteria which doesn't have, and get incorporated into the DNA of the next bacterial cell in which it has entered by means of a bridge which is called as your conjugation bridge. Now suppose if this is a bacterial cell and this is another bacterial cell. Now the bacterial cell which will be having this plasmid in it, suppose this is the normal chromosome, this is the normal chromosome, it does not have this extra chromosomal DNA. This one is called as your plasmid. This is called as your plasmid. Okay, and this is its normal DNA. This is the normal DNA. So we call this type of bacteria to be the F plus, and this one is to be the F minus. And these bacteria which are F plus, they have certain projections on their cell wall. We call them as your sex pili, and with the help of this sex pili, they can build a kind of conjugation bridge between the two bacterial cell. And when this bacterial cell produces this conjugation bridge, this plasmid at first it undergoes replication. It undergoes replication, and one of the replica goes and it will now bind with this that is the bacterial cell which did not have this plasmid. Now since there is a recombination of the genetic material between these two bacterial cells, we can say that this is a kind of sexual reproduction in case of your bacteria, and this type of sexual reproduction is known as your conjugation. Okay, there are three types of sexual reproduction. We are not going for that because it is not in our syllabus. Just for your information that during unfavorable conditions, sometimes the bacteria also reproduces by means of a sexual reproduction. That is a process called conjugation, when they have this extra chromosomal DNA along with that of the normal DNA, which at first replicates, then passes through the conjugation bridge and incorporates into the DNA of the other bacterial cell. So since there is a genetic combination, so we can call this type of reproduction to be a sexual reproduction. Here gametes are not formed, okay, no gametes are formed, just exchange of genetic material and incorporation since it is taking place. So definitely the characteristic of this bacterial cell is going to change. So that is why we call it to be a kind of sexual reproduction in case of bacteria. But normally it reproduces by means of your asexual reproduction. Now, along with the appendages, which was called as your sex pili, which determines the sexual reproduction in case of bacteria, sometimes the cell wall of the bacteria is provided with that of a locomotory organ, which is called flagella. Now, this flagella can be single. This flagella can be in the form of tuft. This flagella can be throughout the cell wall of the organism. So depending upon that also bacteria is classified. But here we are not discussing the types of the bacteria depending upon the number of flagella or the location of the flagella present on the cell wall. What you should know at this point is that the characteristic feature of the eubacteria is that the one which are motile, they have flagella as their locomotory organ. And other than these extensions, they have some of them, they have the sex pili in them, which helps in the process of sexual reproduction, that is conjugation, by forming the conjugation tube in case of the bacteria. Besides, some of the bacteria sometimes have some bristle-like structures on their cell wall. Bristle means hairy structures on their cell wall. Now, these bristle-like structures are known as your fimbriae. Fimbriae means finger-like projections. Now, fimbriae or fimbriae as it is called, it is mainly meant for the bacteria to stick to some kind of substratum. It helps it to hold into some substratum. So, these are the various kinds of extensions which are found in the cell wall of the bacterial cell. The next thing, as I was telling you about the gram-positive and the gram-negative bacteria, now it is depending upon the staining technique, isn't it? But then why is it that the gram-positive is able to hold the stain and the gram-negative is not able to hold the stain? The reason is because the gram-positive bacteria's cell wall is comparatively thicker in compared to that of the gram-negative bacteria, and also the gram-positive bacteria have in their cell wall, along with the peptidoglycan, teichoic acid. This teichoic acid or teichoic acid, as you may pronounce it, is absent in case of the gram-negative bacteria. So it also helps to retain the stain compared to that of the gram-negative bacteria. Then regarding the cyanobacteria which I was talking about, some examples of the cyanobacteria are nostoc, anabina. It also lives in symbiotic association. They have their body is filamentous, and in between the filaments they have a structure which is called heterocyst. Now, heterocyst, when you color them, all other compartments of the filament will take up the color, but heterocyst remains hyaline. It does not take up color, and it is also associated with with the enzymes which can fix the nitrogen. So it helps in the nitrogen fixation. So nostoc and anabina helps in they can be used as biofertilizers, and they are cyanobacteria. So that was the thing. And heterotrophic bacteria, I told you that what they are. That means they cannot prepare their own food. They are either saprophytes or they are parasites, or they are also acting as your symbionts also. Okay. So this was about your eubacteria. And archaebacteria, I told you that depending upon the kind of locations they are found, that is in saline halophiles they are called. Thermoacidophiles means they can withstand the extreme temperature as well as extreme acidic condition of the soil. So these are all because of again the difference in the cell wall and the cell membrane composition, which makes them means a strong group of your bacteria belonging to the kingdom Monera. The next thing is about your mycoplasma. Now, mycoplasma are the smallest living organisms so far found, and they are associated with causing some kinds of diseases in case of domestic animals, and they do not have cell wall. They do not have cell wall, which is the characteristic features of the organisms belonging to kingdom Monera, having a cell wall, but they do not have cell wall. Then why are they included here? The other characteristic matches with that of the kingdom Monera. That means they will have the DNA which is lying freely within the cytoplasm. They have the 70s type of ribosomes. They have the mesosomes. Now what are mesosomes? Mesosomes are simplified, you can say, organelles found within the that is a cell of a bacteria, which are having the enzymes responsible for respiration. So respiratory enzymes are found over there. They have cell inclusions. Their reserve food material in case of kingdom Monera is your glycogen. Okay, they have glycogen as the reserve food material. And in case of that is your cyanobacteria, it is your starch. Okay, that is the reserve food material in case of the cyanobacteria, because they are capable of doing photosynthesis. So this is all about the characteristic features of the organisms belonging to kingdom Monera. Economic importance. Economic importance means how they are useful to us and how they are harmful to us. So in every kingdom, no, we are going to discuss about the economic importance. So usefulness is that organisms belonging to eubacteria, they are used for making antibiotics. Antibiotics, you might have heard about penicillin, which is formed from fungus. It is not from bacteria. But there is a group of bacteria which belongs to streptococcus. So there is a antibiotics which is called streptomycin. So streptomycin is formed from Streptomyces bacteria. Okay, Streptomyces griseus. So that is the bacteria from which the streptomycin is formed. Then it is used in making curd. This is very common thing. We are using it in our everyday life. Lactobacillus group of bacteria. We call them as lab bacteria. That means lactobacillus acid bacteria. So they are togetherly known as your lab. So curd is formed as a result. This is useful for our body. They form probiotics. Useful bacteria are there in the alimentary tract, which help in the process of digestion as well as assimilation of some vitamins in our digestive system. They help in the process of formation of vinegar. Okay, Acetobacter aceti, it helps in the formation of acetic acid, which is nothing but vinegar. Then biofertilizer, just now I told you nostoc and anabina, they have heterocyst, and the heterocyst have the nitrogen fixing enzyme in them, so they help in as biofertilizer. Biogas. Now biogas is formed from this archaebacteria. There is a certain group of bacteria which is called methanogenic bacteria. Now this methanogenic bacteria are found in the gut of the ruminants, that means the grazing animals, the herbivores, and as a result of that is the dung of the animal. It contains those those bacteria, that is methanogenic bacteria, and it helps in the formation of the biogas. So this is economically important. It helps to serve the purpose of energy. Manure, since they are decomposers, so they form good manures which can be utilized for raising up the crops. Food supplements. There is a organism called spirulina. Now spirulina is used as protein supplement. Okay, now that what does it do is that it also helps to reduce weight. How does it do so? It enhances the metabolism of our body. If you take the tablets of spirulina, so that is they are used as supplements, protein supplements, and also they are good in terms of increasing the metabolism of the body. So these are the various usefulness of the organisms belonging to kingdom Monera. Harmfulness. What are the harmfulness? Of course, the diseases of human beings, crop plants, animals, that is caused by that of most of the organisms belonging to kingdom Monera. You might have heard about cholera, isn't it? Vibrio cholerae causes cholera. So then you might have heard about pneumonia. Then you might have heard about tetanus. These are the diseases of human beings. Similarly, you might have heard in case of that is your cow, that is the domestic animal. They suffer from the disease called anthrax. And so various kinds of animal diseases, various kinds of human diseases, fowl cholera, that is the cholera which the that is poultry birds suffer from, that is also a kind of bacterial disease. So these diseases can be prevented by taking that of again the antibiotics, and of course by taking some other drugs. So this is how we can get rid of. And crop plants are also affected by that of the bacterial diseases. Like you might have heard about a disease called citrus canker. It is mentioned in your book, citrus canker. It is caused by a bacteria, Xanthomonas citri. Okay, Xanthomonas spelling will start with X, X-A-N, Xantho, T-H-O, tho, Xanthomonas, M-O-N-A-S. Okay, Xanthomonas citri, C-I-T-R-I, citri or citri, whatever you want, you can pronounce it. So that is the generic name and the species name. So that causes the disease called your citrus canker in case of the, that is your lemon family fruits, plants. Then of course crop plants also bacterial blight, that is in case of rice plant it takes place, in case of the wheat also it can take place. So they affect our crops, so production is affected. So that is the harmfulness of the bacteria. Spoilage of food. Bacteria love to grow in the places where there is water, where there is protein and the temperature is 37 degrees Celsius. So if you give them all these right conditions, they can grow there and they can spoil your food, and of course degrading them into simple compounds which can be harmful. If you take it, you may fall ill. Okay, so these are the various harmfulness of the organisms belonging to kingdom Monera. So that's all about the various studies related to the various categories of organisms belonging to kingdom Monera, their characteristic and economic importance. That's all for today's class. I hope you have understood the class well.